Hello everyone. So today I'm going to discuss about the drug extraction from natural products. So role of natural products in drug discovery. So let us see. So natural products are characteristic of high chemical diversity, biochemical specificity, and other molecular properties that make them favorable for drug discovery. So types of sources of natural products for drug discovery. So these are the pictorial representation sources of drug from various natural products. So you can see microbial world, animal sources, marine world, venom and toxins, and plant kingdom. The sources of natural products for drug discovery. So we can say animal sources. What are the common drugs we extract like insulin, thyroxine, and plant sources like vincristine, pilocarpine, microbial sources like penicillin, streptomycin, and marine sources like avarol, which is reported to be an influenza inhibitor. So preparation of initial extract. So there are various way how to prepare the extraction of natural products to extract the drug molecule. So first, extraction means the separation of medically active portion of the plant or animal tissues from the inactive or inert component by using selective solvents. So extract can be defined as preparation of crude drugs which contain all the constituents which are soluble in solvents. So there are various method of extractions like infusion, decoction, maceration, percolation, or reflux extractions. So what is infusion? So fresh infusions are prepared by macerating the crude drug for a short period of time, which with the cold or boiling water. So these are dilute solution of readily soluble constituent of the crude product. So what is decoction? The decoction is a method of extraction by boiling herbal or plant material to dissolve the chemicals of material which may include system, root, bark, or rhizome. So in this process, the crude drug is boiled in a specific volume of water, like 1:4 for a defined time, and volume is reduced to 1.4 of the original, and it's then cooled and filtered. It is used for water soluble and heat soluble constituent like tea and coffee. Maceration. So in this process, solid ingredients are placed in a stoppered container with the whole of the solvent and allowed to stand for a period of at least 3 days, sometimes 3 to 7 days it's required, with frequent agitation until soluble matter is dissolved. The mixture is then strained through sieves, nets, and mark pressed and combined liquid clarified by filtration or decanation after standing. Percolation. So, what is percolation? It is continuous downward displacement of the solvent to the bed of crude drug material to get the extract. So, you can see from the picture how it could be extracted by percolation method. So reflux extraction, which is one of the common what we do in the laboratory. So reflux extraction is a solid liquid extraction process at a constant temperature with repeatable solvent evaporation and condensation for a particular period of time without the loss of the solvents. So there are now a lot of modern method for the extraction of the drug molecule. So the most useful is Soxhlet extraction, then pressurized liquid extraction, supercritical fluid extraction, ultra sound assisted extraction, microwave assisted extraction, pulsed electric field extraction, and enzyme assisted extraction. So, what is Soxhlet extraction? So, Soxhlet extraction is used where compound have limited solubility in a solvent and impurity in the insoluble in solvent. So, solid material containing some desired component is placed inside a thimble made from a thick filter paper, which is loaded in the main chamber of the Soxhlet chamber. The extractor is then placed in a flux containing extraction solvent. Soxhlet is then equipped with the condenser. So the solvent is heated to reflux. Then the solvent vapors travel up the distillation arms, condensed by condenser, and travel back to the chamber holding the thimble of solid. The chamber filled with the warm solvent and dissolved some of the desired component. The chamber is automatically emptied down the distillation chamber by siphon side arm. So pressurized liquid extraction. So pressurized liquid extraction is a sample preparation technique that combines elevated temperature and pressure with liquid solvent to achieve fast and efficient extraction of the component of a solid matrix. So PLE applies up high pressure in extraction. High pressure keeps solvent in a liquid state above their boiling point, resulting in a high solubility and high diffusion rate of liquid solutes in the solvent and high penetration of the solvent in the matrix. So, pressurized liquid extraction dramatically it decreased the consumption of extraction time and solvent and had better repeatability compared to the other methods. So this is the commercially available pressurized liquid extraction unit. You can see. Uh, Now, supercritical fluid extraction. So this uses solvent that are kept in a physical state known as supercritical as the extraction solvent. They are formed when the solvent is kept above its critical point temperature and pressure. A supercritical fluid normally has the density and appearance of a liquid, but its viscosity is lower than would be expected for a liquid. So supercritical fluid are used for extraction because the diffusion coefficient of solute and higher than would be normally be accepted for a liquid. Consequently, their penetration and mass transfer of constituents from the natural material is usually good. The extraction rate in the case of solid is governed by particle size. The smaller the particles, the better the rate of extraction. Liquid or wet natural material samples are often absorbed in inert drying agent such as magnesium sulfate prior to extraction. So this is a schematic representation of an offline super critical fluid extraction. So you can see supercritical fluid reservoir is there, then pump, and then is a valve, then the solvent is passed to the chamber, and then we can collect the material at the bottom, collection vessels. Supercritical carbon dioxide is used to extract volatile oils, lipid, uh, then terpenes, steroids, alkaloids, and carotenoids from natural materials. It is used on an industrial scale to extract bitter acids from hops and to remove caffeine from coffee beans, avoiding the use of environmentally harmful solvents. Supercritical fluid extraction has also been used to extract the anti-cancer drug Taxol. Okay, so this is the structure of the Taxol. The alkaloid vindolinine from Cerathosas rosaceae and parthenolite from feverfew. So this is the structure of A is the structure of Taxol. The B is the structure of vindolinine, and C is the structure of parthenolite, okay, which are extracted from by supercritical carbon dioxide method. Ultrasonic and assisted extraction. The procedure involves the use of ultrasonic waves, which have frequencies higher than 20 kilohertz, have a great effect on extraction yield and kinetics. So EOE in so the ultrasonic is an assisted extraction involves ultrasonic effect of acoustic cavitation. Under ultrasonic action, solid and liquid particle are vibrated and accelerated, and because of that, solute quickly diffuses out from the solid phase of the solid phase to the solvent. So merits to increase the of extraction yield and faster kinetics, reduce the operating temperature, allowing the extraction of thermomobile, labile compound. Demerits the active constituent of the medicinal plant to formation of free radicals and consequently under undesirable changes in the drug molecule. This is the picture of classification one. Enzyme assisted extraction. The structure of the cell membrane and the cell wall micelles formed by the macromolecules such as polysaccharides and protein, and the coagulation and denaturation of the protein at high temperature during extraction are the main barrier to the extraction of natural products. The extraction efficiency will be enhanced by enzyme assisted extraction due to the hydrolytic action of the enzyme on the component of the cell wall and membrane, and the micromolecule inside the cell which will facilitate and release the natural product. Pulse electric field extraction. This is another modern technique. So pulse electric field is the application of a short time pulsed with the high voltage into the food product placed between two electrodes, thus promoting the modification of membrane permeability and increase the extraction yield. Recently, PEF treatment has been applied in order to recover pigment from the beetroot. So pulse electric field extraction significantly increases the extraction yield and decrease the extraction time because it can increase mass transfer during extraction by destroying membrane structures. Microwave assisted extraction. So microwave generate heat by interacting with the polar compounds such as water and some organic components in the plant matrix following the ionic conduction and dipole rotation mechanism. So yeah, this is the pictorial schematic representation of a microwave and how we can set up. So when you set up, just it can hit the solvent and it can extract the compound. Preliminary biological screening. So biological screening is used to detect the presence of active constituent in the initial extract. So screening of natural products and their extracts is complicated due to the presence of fluorescent and insoluble compounds. So advances in detection technology and biological screening assay have overcome this problem. Method of biological screening. So first one is high throughput screening. So in this process, a large number of crude extracts from the organism can be simultaneously evaluated in a cell or a non-cell based format, usually utilizing multi-well micro titer plates. So using robotics, data processing and control software, liquid handling devices, and sensitive detectors, high throughput screening allows a researchers to quickly conduct millions of chemicals, genetic or pharmaceutical, pharmacological tests. So biochemical target test compound and then compound with a desired effect on a heat. So this is called high throughput screening, chemical representation, schematic representation. As a random screening. So this approach strives to find among a great number of molecules, several hundreds of thousands, on that could be active in a given indication. So in this case, therapeutic objective is fixed in advance. So great number of molecules is tested, but on a limited number of experimental models. Major discoveries by this process. So, what are the major drug molecules has been discovered by this process? Like Taxol, anti-cancer drug, we all know, streptomycin, okay, artemisinin, and then statins, and the morphine. So, these are the natural product extracted from the natural sources. The virtual screening. So virtual screening is a computational technique used in drug discovery to search libraries of small molecules in order to identify those structures which are most likely to bind to a drug target, typically a protein receptor or enzyme. So what are the merits of this technique? So it's like computational, only high scoring ligands and goes with assays. And demerits molecular complexity and diversity, sometimes false positive signal it gives, and synthesis is it cannot synthesize, it can just predict. So phytochemical screening, so it refers to the extraction, screening, and identification of the medicinally active substances found in medicinal plant. The Phytochemical research approach is considered effective in discovering bioactive profile of plants of therapeutic importance. So phytocompounds such as the alkaloid, tannins, saponins, phenols, flavonoids, and terpenoids are observed to be present in the investigated plant in variable proportions. Methods of compound purification. So purification is the physical or chemical process of removing contaminants from a compound. The purification method mainly relies on chromatography, and the final product is obtained through crystallization. The physical process include like sublimation, distillation, crystallization, and chromatography. These are the technique we can use to purify the extracted compounds from the natural products. So what is sublimation? So sublimation is a transition from solid phase directly to the gas phase without passing through the intermediate liquid phase. So you can see we can take the solid material and we can heat it and we can just sublime like it can, we can convert it to gas phase, and then the gas phase can be condensed collecting flux. We can get the pure product. Distillation. The distillation is a process of separating the components or substance from a liquid mixture by using selective boiling and condensation. Fractional distillation. This is very useful method for purification. So fractional distillation is a separation of mixture into its component parts or fractions. So chemical compounds are separated by heating them to a temperature, and which one or more fraction of the mixture will vaporize. Crystallization is one of the common technique. So it's a purification technique to separate solids from a solution. So we can take a mixture of components solution, then particularly one of the material will crystallize, then filter it, and then wash it and get the pure product. So this is the crystallization technique. And then chromatography, widely used in the organic synthesis. The chromatography is technique for separation of components or solutes of a mixture of basis of the relative amount of solute distributed between the moving fluid stream, called the mobile phase and the stationary phase. So you can see we have a stationary phase where we have to load the compound, and then by passing through solvent we can separate based on their polarity. Then finally, uh, structural elucidation and identification of the compound. So whatever we got pure product. So structure elucidation in the drug development is the process of determining the chemical structure of the compound from the identification and characterization of novel impurities, degradants, and metabolites across raw material, API, formulated products, or biologically metrics. So structural elucidation of organic compounds will often involve the use of NMR, nuclear magnetic resonance spectroscopy. Other identification technique include mass spectroscopy, infrared spectroscopy, and X-ray crystallography. So after we get confirmed structure by this technique, we can use them as drug molecule. So thank you everyone for listening.